All right, so today I'm really excited to reintroduce Dr. Sam Scarpino, who, as you may remember, was our inaugural speaker in this Colloquium series, and fittingly is our first returning one. As you all are probably familiar with Sam from his first presentation, I'll just give a shorter introduction with my perspective, because I think Lauren introduced him in the beginning. So even though Sam graduated from Lauren's lab a few months before I started, I've really always seen him as a role model and mentor. And from the beginning, he kind of helped me learn and get my feet under myself. So first it was about Austin, and he showed me some of the fine establishments downtown and around the city during my graduate school recruitment weekend. And this is really random, but he also taught me how to think about the weather in Texas as a Midwesterner. And for some reason, it's always stuck with me. And I hope I am not remembering it wrong, but the quote from him, which I hope isn't completely incorrect, was 70% chance of rain in the Midwest means there's a 70% chance it rains everywhere. But a 70% chance of rain in Austin means there's a 100% chance that it rains in 70% of the city. And I hope I'm not getting that wrong. And that has been, I think, over the past 7 or 8 years that I've been in Austin, completely my experience. So it's pretty funny. Anyway, I also learned a lot from him about science. I've always admired his dedication to open science and collaboration. And, really in my first semester in grad school, he dragged me through my first ever computational project where we were working to predict seasonal flu epidemics. And I learned even more as I did a small part on a collaborative project he led to assist during the 2014 Ebola outbreak. Over time since then, I've watched and been excited as his career progressed. First as an OBDR fellow at the Santa Fe Institute, then as an assistant professor at the University of Vermont. He became an ISI fellow for his work on complex systems. And then he moved to becoming an assistant professor at Northeastern. And most recently, he was named as an external faculty member at the Santa Fe Institute. His work, as you all know, is always on the very cutting edge of complex systems, network science, epidemiology, and evolutionary biology, which is why we've asked him to come back today to discuss his recent preprint looking at the spatio-temporal dynamics of the novel COVID-19 variant in the UK. And so with that, I will let you take it away, Sam.
Great. Well, Spencer, thank you so much for the very kind introduction, especially probably leaving out some of the more embarrassing details of downtown Austin. But no, I was actually just thinking about some of the flu forecasting stuff. Actually last week, so I'm working with Mass General Hospital and the Brigham to forecast inpatient hospital demand, and came across some code that you and I and others, some of you on this call had worked on previously, and was remembering fondly back, I guess not that long ago. But no, so thank you very much for the introduction and for inviting me, and Lauren as well for organizing. It's great to see everybody here. Certainly wish we were in person, but am still enjoying being able to see some friendly faces. Okay, so today I'm going to, and I assume everybody can see the slides. Great. So I'm going to talk about work we've been doing trying to understand the epidemiology of B117 in the UK. And I will tell you that I still am convinced that I don't understand what's going on. And that doesn't necessarily mean much. But part of the reason I'm excited to tell you all about what we've been doing is because I am genuinely puzzled about some of the patterns that we've been seeing. And I'm sure that many of you on this call will have some great insights. And I think, one of the things that we're going to be dealing with for COVID-19, probably for the foreseeable future, is trying to quickly sort out what's going on with these variants. And I think that kind of pushes a lot of the methods we've been developing, a lot of the ways we've been thinking about infectious disease epidemiology, kind of to the limits of what we may be able to do with early data, and the need to bring together lots of different data sets to try to understand these different variants. And certainly also from an evolutionary biology perspective, I think there's quite a bit of work that we can bring to bear on this. Just as kind of the motivation, one of the patterns that continues to jump out at me is just how much heterogeneity there is in COVID-19 and how it persists across scales. So this is just a global map that I pulled from October of cases by country. And you can see some countries, as we know, have kept it out or eradicated after it showed up. Others, it continues to persist. Similarly, across the US, although some of this has been homogenized of late, but we see quite a bit of variation across space and time in terms of, especially in the spring wave, where there were COVID cases. In Massachusetts, we see a similar amount of kind of heterogeneity, and then the same as we zoom into Boston. And some of the determinants of this heterogeneity cut across scales, issues of racism, socioeconomic inequalities, but mobility patterns, now vaccination coverage, et cetera, all kind of interacting. But of course, there are also important differences in different locations at the finest granularities, but also as we kind of zoom out and look at differences between states and across countries internationally. And this was actually the motivation behind the first paper that we published. And I'm not just putting this up here to kind of remind everybody that we published this paper, but it's because, to me, this is almost exactly what seems to be happening with B117 in the UK. And so just to orient you, the top chart are data that we pulled for real-time mobility in China out of Wuhan into neighboring provinces outside of Hubei province. So the y-axis is just some obfuscated mobility index. So higher is more mobility. I think it's almost certainly not linear, the scale, although they don't tell us what the scale is. And then the x-axis is time going from January 1st through the end of the month. In orange is 2020, and in gray is 2019. And the couple of things to orient you is that there's this gap right before the cordon sanitaire. And because it's nonlinear, we think that that gap is actually quite large. And if you remember back to the quote from the mayor of Wuhan saying that he thought 5 million people left the city in the 72 hours between the announcement of the cordon sanitaire and when it went into effect. And then this really precipitous drop in mobility, basically all the way to zero going into the Lunar New Year, and then persisting for quite a period of time. And on the bottom here, we just have a heat map of mobility out of Hubei and Wuhan into neighboring provinces, just kind of seeing the mobility shrink. And so why am I showing you this? Well, one of the things that seemed to be the case is that a lot of mobility out of Wuhan was feeding cases throughout China, and that when you put the cordon sanitaire into effect, the growth rates drop precipitously. And this is what we see for B117 in the UK. So here I'm just showing you the COVID-19 growth rate at the province level. So each dot is a province. Positive is more growth, negative is less growth. This is like the little r instantaneous growth rate. In the week January 2nd through the 8th, so basically COVID is not growing yet. We weren't even calling it COVID then. And then on the x-axis is the connectivity to Wuhan, so that migration index prior to the cordon sanitaire. We just take kind of a median over this window. And so you see that the growth rates are starting to pick up, and you see there's this kind of like elevation in the points. And it becomes more pronounced as we move into the week just prior to the cordon sanitaire, where there's this very strong positive relationship between connectivity to Wuhan and the growth rate at the province level. Then the cordon sanitaire goes into effect, and you see this line kind of tilting back down again, eventually becoming quite strongly negative. So the places that previously had the most connectivity to Wuhan and the highest growth rates now have the largest negative growth rate. So the curves are coming down the fastest. Now, of course, there's some confounder here, because you imagine that if you go up really fast, you might come down really fast. But one of the things that continues to jump out is this observation of mobility playing a really key role in moving COVID-19 around, especially from places that are having locally large outbreaks. And this was a paper that caught my attention. It's a preprint, but they're just looking at different lineage introductions into Scotland and estimating that there are 300 separate introductions that sparked some of the later waves of outbreaks in Scotland. And so each of these lines are just introductions coming in from Northern Ireland, England, Wales, and other places. And it continues to be this pattern, I think, that we're seeing, that you have lots of introductions that are driving local outbreaks. And it, I think, becomes a little bit difficult to disentangle how much of the growth in a particular area is due to endogenous transmission versus continual reseeding, and then some of these transmission chains going locally extinct. So whether we really should be thinking about this, and of course this is my perspective on it, and so I'm biased, but whether we should be really thinking about this as more of a meta-population model with lots of sort of source sink dynamics, as opposed to many locally stable outbreaks that are kind of self-sustaining if you were to turn off those reintroduction of cases from the meta-population. And this was motivating or potentially biasing, depending on whether you think we're generally correct about these meta-population dynamics, the work that we've been doing on the B117 lineage in the United Kingdom. I should say I'm mostly going to say United Kingdom, but the bulk of the data we have access to are actually from England, and so quite a bit of what we're talking about is really only relevant for England and not Wales, Scotland, Northern Ireland. And this is work that involves, as you might imagine, a huge number of people. Verity Hill from Andrew Rambo's group in coordinating the genome data. Moritz Kramer is kind of one of the key leads on the project, coordinating the whole initiative, working with Professor Ali Pybus, and then the COVID-19 genomics working group. So in the end, we're working with a variety of different data sets from the 200,000 or so genomes, the PCR testing data, which I'll explain on the next slide, the local case data, and then local mobility data from static snapshots from O2, which is a data provider, and then real-time weekly mobility data from Google that we're parsing. So we end up with a pretty large number of large data sets that we're trying to line up to make sense out of what's going on. And I think ultimately this is the pattern that we're all trying to wrap our heads around. So this is April through basically a few days before the end of January. The proportion of cases in the UK, again, mostly England, that are positive on a PCR test for this S gene target failure. And so one of the things that Public Health England and others figured out right away is that there were lineages that had deletions in the spike gene that were causing primers to fail on the PCR tests. And so they actually have a concerted effort to test between 50 and 80% of all the cases in the line list using a primer set for the S gene, and they report on the proportion of cases where the primer for the S gene fails to bind. Now, running into December, B117 was a minority lineage that was carrying this S gene deletion. And so if you look kind of prior to December, the bulk of these are not B117. In fact, prior to kind of September, October, probably none of these were B117. But then, as I'll show you, moving into December and January, B117 comes to dominate all of the lineages that are carrying this S gene deletion. And so towards the right-hand side of this curve, most of these cases are B117 or are thought to be B117. And so essentially what we're trying to understand here is that we have these S gene target failures that we're making up, somewhere between 5 and 10% of all the cases, kind of, wobbling around. And then in late November, marching through to the present, we go from 5 to 10% of cases up to 75+%, 80% of cases, which is, pretty spectacular and obviously very concerning. One of the first things we wanted to try to understand was the bias that might be present in these S gene target failure results. And so I apologize, this slide is a little bit busy. I'll walk you through it. So one of the cool things we can do with the genome data is we can look at the actual B117 frequencies. We can also pull all of the lineages that are known to carry the deletion. And so we can come up with a proportion from the genome data of B117 and a proportion from the genome data of all lineages that are carrying the deletion. And then I basically just subtract what the S gene target failure proportion is for a particular time window from what the genome data tells us the proportion of B117 should be, and I do the same for the deletion. So the red on the left is S gene target failure proportion minus B117 in 5 different time bins. The right is the S gene target failure minus the deletion across all the lineages in 5 different time bins. We've creatively named, originally the time bins were early and late. They have since internally become early, late, later, latest, and January because we started this back in December. So the first time bin is before the November lockdown, so pre-November 3rd, I think. The second is during the lockdown, so basically the month of November. The third is post-lockdown, so the first 2 weeks of December. The fourth is the second 2 weeks of December, and then the last is post-December, so all of January. And what I want you to notice is that basically the gray is centered on 0 all the way through until the last time bin, where a strong positive bias shows up. The red starts out with a positive bias. So the S gene target failures are overestimating the frequency of B117. And as we get into the post-lockdown period, it's noisy, but it's much closer to being centered on 0, suggesting, as we can see in the genome data, that almost all of the lineages carrying the deletion that aren't B117 are gone. Now, one interesting thing to point out, and we think we understand what's going on here, is that there's this consistent positive bias in the lowest bin. And what we think is going on is that the genome centers actually have some information on whether these genomes may be carrying the S gene deletion or not. And they're targeting their early sequencing efforts to try to find variants that they don't have a lot of information on. And so they queue up the sequencers to look for the rarer variants first based on whatever information they have. And so the B117 sequences actually come off the genome sequencers later. And that we basically, as we roll forward in time, we consistently see this last bin being biased. And we think that has to do with the order in which they do the sequencing of the genomes, and it's not a real bias. Although you may notice in that post-December, post-16th December, there is a bit of a positive bias as well. However, for the most part, using the genome data instead of the S gene target failures, we see the same basic trend. So now we have 5 different time bins, and we have 5 different time bins because the genome sequencing kind of happens in fits and starts, and so we have to aggregate across. Even though they have 200,000 genomes, we have to aggregate in time to get an estimate of the frequency. Each line is a UTLA in the UK. So if you're familiar with the UTLAs, you know what I'm talking about. If you don't, I hope you never have to figure this out because it's a mess trying to line up all of the different UK regions. But essentially, it's like a municipality level. So London is like its own, or Inner London and Outer London are 2 different UTLAs. And then over time, and you can see that in most of the UTLAs, you get this like very rapid growth in the proportion. Although one of the things that's true about the genome data, the growth rates are not as spectacularly high in most places as they are with the S gene target failures. And we also do see some UTLAs where the growth never really picks up, where even it comes down a little bit over time. One of the big challenges here is that we don't really know how the genomes get selected for sequencing. And unfortunately, and this is like the typical stuff that happens with COVID, starting exactly at the time that the S gene target failure frequency shot up, Public Health England went from sequencing 80% of the line list to 50% of the line list. And it's not clear how they pick what individuals are going to get PCR tested. So there are definitely differences between how the sampling happens for the genome and how the sampling happens for the S gene target failure. And we have as yet been unable to really pull apart what's going on with some of these differences. But overall, the pattern is the same, that very rapid rise in B117 and S gene target failures. However, definitely stratified by geography. So not every place that has the lineage ends up shooting up. So just because you may be interested, so what we're doing here for the B117 frequencies is that we define a time region, let's say 2 weeks. We take all of the genome sequences for those 2 weeks, and then we have a list of the ones that are B117, and we have like a simple Bayesian updating scheme for getting a posterior distribution over the proportion of individuals that are B117 positive. So just imagine that I have N sequences collected in UTLA I, and X sub I of those are B117. And so I have some binomial sample that I can use with the appropriate conjugate prior in order to get an estimate of the posterior distribution in each time window. And what I'm showing you here is just the evolution of these posterior distributions in time from earliest pre-lockdown in blue up through January in red. And I just cherry picked a few UTLAs to show you the different kinds of patterns that we see. So in the top left, this is one of the UTLAs in England where you see this like spectacular rise where it basically goes from 0 to 75% frequency over the course of a few months. We see in Scotland, this in the bottom left here, the S is for Scotland, the E is for England, this kind of pattern where it stayed low and then shot up in January. And then we see some other cases, and this is not uncommon either, where just given how rare the sampling is, you get these really spread out posterior distributions where there's not a huge amount of statistical support for this very large, rapid increase in the frequency. Okay, so here are the things that are really confusing us about this. So now that I hopefully have oriented you and not just confused you, the first thing is that B117 arose and started spreading in a part of Southeast England that previous to B117 had had low attack rates. So what we're looking at here are 4 different groups of UTLAs of geographic regions. Purple are the UTLAs where B117 was detected by genome sequencing prior to November. Blue is where it was detected during the lockdown, so in November. Green is where it was detected in December. And black is where it's never been detected, but we just call it unknown because that means there hasn't been any sequencing from those UTLAs. The y-axis is the relative attack rate prior to November 1st, so just the total attack rate prior to November 1st. And I make it relative by dividing every UTLA attack rate by the median attack rate in the unknown set of UTLAs. So if you see the black on the right, the dashed line is at 1, which is the median for the black bin of UTLAs. The purple is the median in those UTLAs that had B117 early. So the median attack rate in the early UTLAs is about 30 to 60% lower than it is in most of the rest of the UK and England. And many of the models that we've seen people estimating the growth rates from, especially those S-curve logistic estimates, are actually not accounting for these differences in initial conditions because of the way they end up doing the relative growth rate calculations. Some of them are. It's a little bit hard to figure out exactly how they're doing it, but some of them are trying to account for differences in initial conditions. But I'm still not fully convinced that anyone has come up with growth rate estimates that really account for the fact that some of these places, and the ones in purple, these are the UTLAs that also had this really spectacular rise in the frequency, had quite low attack rates relative to lots of the rest of the UK prior to the lockdown. Now, fast forwarding to January, let me just explain this and I'll take questions. Fast forwarding to January, we'll do the same scheme. So we have the same bins. So the purple UTLAs are the same as they were in the previous slide, the same with the blue, the same with the green. However, now I'm calculating the attack rates up through the end of December. So we've gone through these December waves of B117, and I'm again dividing everything by the median of the attack rates in the black, the unknown, or the rest of the UK. And what you can see is that the purple has come up to match the rest of the UK. And so basically what happened is the purple UTLAs had low attack rates. B117 emerged there. They had huge outbreaks from B117, and the attack rates mostly equalize across all of the UK, aside from places where it was detected post-lockdown that end up actually having relatively lower attack rates. Questions?
Yeah, I was wondering, how is the attack rate measured? Is it just reported cases, or is it serosurvey, or is it something else?
Yeah, so that's a good point. It's just reported cases, and so there are tons of reasons why this may not be the right, or this is certainly not the right way to do this. Maybe I can wave my hands and say that the errors should be relatively similar across all of the UTLAs enough so that this normalization tells us something relevant in terms of the relative, but it also could be the case that surveillance was not as good in Southeast England, and that this is totally an artifact. But yeah, I think it's a really important thing to follow up on, is that I'm just taking the reported cases and using that to calculate the attack rate.
So taking a step back, what's the take home from this graph if we believe, you know, the data are good?
Yeah, so the take home is B117, the places where it had the, and so I haven't shown you this yet, but the purple is where it had the highest growth rates, where it emerged early. They had 30 to 50% lower attack rates, meaning substantially higher fraction susceptible during that window of time when the B117 wave swept through them. And now we go into January and the growth rates, and I'll show you this in the next slide, and all these purple locations are crashing much faster than neighboring locations. And if you were to just plot like fraction susceptible, you see it starts out much higher in purple and then drops down to match everybody else as you go into January. And that very closely tracks the growth rate differences that we see in these different locations. So one of the things that I think we need to account for in the growth rate essay, I think there's probably still evidence that the growth rates are higher for B117, but we need to really account for the fact that if these data are correct, there are much higher fraction susceptibles in these places with the high B117 growth rates.
Well, another factor that would affect the different, you know, the non-logistic growth in some areas is not having a mixed population. If you've got a bunch of towns, some of which are seeded and some of which are not, you know, if you've got, you know, if they're not mixed, then within each town, the fraction that is B117 is going to grow logistically. But if in some towns the total number of infections starts to saturate, either because you're approaching herd immunity or because the situation is so bad that people change their behavior, then the fraction of B117 in the aggregate of all the towns is not going to grow logistically. It's going to grow slower than logistically. And so that might just describe a difference between Scotland and the south of England.
Yes, I think that's right. Although the I lost. Oh yes. So now I remember what I was going to say. Sorry. So I think that this is also an important point, because to me that the two most compelling pieces of data to suggest that the growth rates are higher for B117 are the contact tracing data that Public Health England has published, showing a higher number of secondary infections when B117 is the index case, and that we're seeing the growth rates stay higher in B117 in the lockdowns compared to non-B117. And importantly, I think because the contact tracing data are reported at an aggregate level, so they're aggregating up from the UTLAs, and we have really high differences even in neighboring UTLAs in terms of the fraction B117 and the fraction susceptible, that the contact tracing patterns could still be explained by the fraction susceptible. I think that's not probably explains all of it, but if you have a higher fraction susceptible, you can have a higher number of secondary infections, even if the growth rates are the same once you control for the fraction susceptible. In addition, and this is, I think, the really interesting point about the mixing structure, one of the things that we did not fully explore, but I mean, some of you have, in particular, some of Lauren's work on the hospital outbreaks in Vancouver, that the mixing structure can affect what happens when you have when you have interventions. And so if B117, for example, has a higher higher number of secondary infections because it has a shorter generation time, for example, that shorter generation time can interact with the way the lockdowns occur to modulate the growth rates as you go in and out of lockdowns. And so that's, I think, something that we need to also be accounting for, is that the UK, during this period of time, the schools opened and closed twice, I think, and they went in and out of a lockdown twice during this period of time when you have the B117 lineage spreading. So you have heterogeneous landscape for attack rates. You have 2 entry and exits from lockdowns and 2 entry and exits from schools, all of which are going to interact with the local population structure, mixing household structure to modulate the actual growth rates that we would observe at the UTLA level or at the aggregate kind of regional level. And so I think that that's part of the reason why really getting an estimate of what the different growth rates are for B117 is really challenging in this environment, because so many things are changing and we know or suspect those things are interacting with the local environments. Okay, so a couple more slides.
Sorry.
Michael. Yeah.
Just one quick question. You're saying fraction susceptible. How is that calculated? Where do you get the fraction susceptible from?
Oh, I'm just using the attack rate minus the population size. So it's, maybe we could just say that's a fantasy and we don't believe that I'm doing that correctly. Yeah. But I think figuring out, maybe from the genome data, you could do it, what the fraction susceptible actually is. Maybe to say it a different way, I think there's some suspect oriented, that's not a word. There's some evidence that should make us wonder whether there are relevant differences in the fraction susceptible in some of these places with really high growth rates that we should follow up on. Maybe that's the way I want to say it. Okay, so just a few more slides. Y-axis is the increase in variant frequency through December on a log scale, and the x-axis is the mobility from London into those UTLAs on a log scale. So each dot is a UTLA. And what we see is this like consistent, we see this pattern consistently depending on how we slice the mobility data and whether we look at variant frequency or growth rate, that you do have this positive relationship between mobility and variant growth rate, but there's this kind of like saturation that is happening. And what seems to be going on, and I have a better slide for this, I just didn't have time to pull it together this morning, is that the UTLAs that are well connected to London seem to basically be lagging London's B117 frequency. So on the x-axis, this is the frequency in London, let's say 2 weeks ago, and the y-axis is the frequency outside of London, present time, or 2 weeks in the future. And the red squares are the UTLAs with high connectivity to London, and the blue dots are with lower connectivity to London. And we see this fairly consistent signal, depending on how we slice the data, that some of this saturation seems to be coming from the fact that a lot of the UTLAs around London are just lagging behind, that they like quickly come up to whatever London was 2 weeks ago over the course of the outbreak. And the way that we've, question, yeah.
Not that it's not direct evidence that the new variant is outcompeting the original variant in, let's say, Oxford. It's that it's outcompeting the original variant in London and then getting imported to Oxford.
That is certainly a hypothesis that we don't think can be completely ruled out, or at least that, yes, basically, that it may have a higher growth rate in London. And I don't necessarily know how much of that we can explain with the local attack rate differences. But yes, that's certainly a hypothesis that we think deserves more consideration, is that it is outcompeting in London, in the London area, and being exported into other places because of the high mobility and large number of cases and large number of people in London. So you think about London and lots of mobility, lots of cases, huge outbreak, just swamping the local outbreaks with B117 lineages. And so just very briefly, the way I classify these red versus blue is I can just run a fairly simple community detection algorithm over the mobility data and identify the cluster of UTLAs that are inside of the London community. So this red community down here at the bottom. For those of you that are interested, it's one of the PhD students in the Network Science Institute, he's now a postdoc, got his hands on this. And so it's actually a little bit more complicated that I'm doing justice, so apologies if you're listening to this in the future, but just community detection. So the red is the red. Okay, last two slides. So now going into January, here on the y-axis is the decrease in growth rates into the lockdown in January. So high growth rates, lockdown in January, highly effective. So more negative means the growth rate's dropping faster. The x-axis is the frequency of the variant of concern based on the S gene target failures prior to the lockdown. So those places that had 75, 80% variant frequency now have the largest reduction in growth rates. Now, of course, if we assume that the curve is symmetric, then if you have a high growth rate up, you will have a high growth rate coming down. So there's some other things in here that we have to control for, but one of the patterns that jumps out is this same kind of pattern that we saw in Wuhan that I think still has a lot of work to explain even there, is that you have lots of mobility from a place with a self-sustaining epidemic and a lot of people that are correlated with the local growth rates, and that when you turn off the mobility and you see these crashes in the growth rates, which I think is consistent with this idea that you have lots of importation that's driving and reseeding a lot of these local outbreaks, making it very difficult to interpret some of the growth rate differences in space, because there's so much happening with respect to the reseeding of the lineages. And then the last thing here is just to say that if I had to kind of describe the pattern of B117 and S gene target failures in one word, it would be mess. So here are two plots, September through February. Each line is a UTLA, and I'm calculating its instantaneous growth rate in weekly rolling windows rolling forward in time. For the non-S gene target failure cases in black, and the S gene target failure cases in red. And if you pick out any one of these lines, so here in December, like just prior to December, this is the lockdown. So there are some places where S gene target failure is got a high growth rate in the lockdown, which is bad and consistent with it having a high growth rate. There are other places where it has a low growth rate, a negative growth rate. And then you move into December, and yes, you see a pretty consistent signal of all of them going up, but not everywhere. And then we get into the lockdown in January, and pretty much everywhere has negative growth rates, but a few places have positive growth rates. And we see a similar kind of pattern in the non-S gene target failure, though everything is kind of depressed a little bit. So this to me is a signal that it probably does have an advantage over many of the non-S gene target failure lineages, because we do see this overall depression in the black curves at the UTLA region, and overall kind of elevation in the red curves. But the curves are kind of all over the place. And I think they're all over the place because you have a heterogeneous landscape of susceptibles. Again, I haven't really shown you evidence that you should necessarily believe me on that, but heterogeneous landscape in terms of susceptibility. One of the major epicenters of the outbreak was in London. So huge amount of connectivity and a huge amount of cases in London. Schools opening and closing 2 or 3 times, lockdowns starting and stopping at least twice, including now, which is a really extreme lockdown. All of this under this landscape that I haven't really talked about yet, which is one of the other models that I'm very curious about, is imagine that every new variant draws its transmissibility from a Gaussian with fixed mean and variance. If you have these lockdowns with lots of local kind of quasi-isolated populations, you will end up sometimes with pairs of lineages that have really different relative growth rates, even though they came from the same underlying growth rate distribution. And that you will see some of that advantage disappear, not all of it, but some of it disappear as it gets out and mixes with the other lineages that have growth rates that are closer to the mean of that distribution. And so I think maybe what's going on here is that B117 had a really big advantage for some reason in Southeast England, got into London, sparked a big outbreak. I think there is evidence that it probably has an advantage, but that it maybe isn't 50% across the board. That it depends on the local population structure, it depends on what other variants are present, all the things that we kind of assume have to be true here. but that if we really are going to try and communicate out to the public and internationally about what these growth rates are, that we need to either more realistically account for all of the things that are changing in time and in space, and that's going to lead to a lot of uncertainty in our ability to really estimate what's going on with these different variants. So finally, just a little bit of speculation, and I would be curious about some of your thoughts on this. So I think the local variant context probably matters a lot, and is something that we do have some ability to assess in the UK, but not really anywhere else. If you kind of squint at the data from Denmark, you maybe can convince yourself that there's some evidence for that as well. But the local variant context probably matters a lot, and it's something that we just almost everywhere have no real ability to deal with. If I had to guess, that the B117 transmission advantage is coming from the fact that it could be a PCR and antibody escape variant. So you imagine that people are false negative if they're carrying B117, and so they're out contacting people. It would explain almost all the data that we've been faced with, including the fact that the lockdowns do seem to work pretty well against B117. I think there are plenty of other explanations. The non-PCR escape variants explanations that I think, and others, especially in the SAGE groups and others, and the rest of the co-authors, I think there's a lot of evidence for is a longer infectious period or a shorter generation time, and those are consistent with the higher viral loads that are estimated in the lab. So if you have the higher viral loads, you may have a shorter generation time or a longer infectious period, and that would also explain the contact tracing data. That would explain why the lockdowns work, because when you go into the lockdowns, you're going to infect everybody in the household, but you're going to stop the infections outside of the household. So you're going to normalize across some of the secondary infection distribution once you go into the lockdown. So you'll take away some of that advantage from B117. So I think those are what some of the evidences are. Again, I want to thank Moritz, who's really driving a huge amount of the work here, and Verity Hill, who is in Andrew Rambo's lab and has been leading most of the work on the genome data, and Professor Ali Pybus, who's kind of coordinating all of us. And of course, if you look in the acknowledgements, there's like literally 10,000 people in the acknowledgements that are working on the COVID genome project. So thanks again for having me. Happy to take questions